My presentation will be a little bit different from the previous concept. My presentation will focus on biomaterial and especially enhancement of biocompatibility of hydroxyapatite with boron, and also I would like to mention a little bit of silver incorporation to hydroxyapatite. And the concept is we try to make the defect chemistry in the structure to obtain some modified quality of hydroxyapatite. So for this purpose, we focus on precipitation. First, I would like to mention about the material, the background of the material, which hydroxyapatite we focus on as an osteoconductive and osteoinductive material, which it can form strong bond with the bone, natural bone, and also it can help bone to grow as a synthetic bone material. And also another element that we use is a boron. It also has beneficial in cellular function with osteoporosis and also some periosteoblast activation as an element. And also silver is known as antibacterial element, which we focus on as antibacterial property in this study. So in general, we try to dope these two elements in hydroxyapatite separately in this, I will mention this presentation, because of the expected boron doping to improve the proliferation and differentiation of osteoblast cells, and also doping of silver into hydroxyapatite to minimize the infection problem. So for this background, we got motivated with the idea of enhancement of osteoblast addition with boron incorporation, and also improvement antibacterial property with silver containing hydroxyapatite. And this is studied before, but the first time we tried defect chemistry for this hydroxyapatite structure that we focused on in this study. So the objective of the study is to synthesize boron and silver containing hydroxyapatite nanoparticle and explore the effect of stoichiometric value of 1.67, which hydroxyapatite stoichiometric value, with different deficiency, which we exchange the phosphate with dopant, and we call it phosphate deficient. Also, and the side of the calcium is a calcium deficient. And we exchange this with different boron or silver with in the solution, in the precursor solution, and obtain enhancement in the bioactivity. So the synthesis of hydroxide on a powder, we follow precipitation method, and these on the left side, the raw powder, we use calcium and phosphate source, and silver source as a silver nitride, and boric acid as a boron source. And on the right side, you can see the follow chart of the precipitation method with mixing the precursor solution in the calcium nitride solution with a whole day of stirring, and then precipitating the nanoparticles. And the important thing, while we follow this chart, we exchange the doping ion with the different deficiency side. So for this purpose, we on the table you can see, for example, the boron with different amount of boron, we decrease the calcium or the phosphate and include the boron ions in the solution. And in the same case, we also did the similar synthesis process with the silver ions, as you can see in the tables. If we follow with these synthesis, we fabricate the green pellets with the diameter and pressure, and then we heat treat it with the range of 500 to 1300 centigrade degree for whole day, and then we analyze this with phase, chemical composition, morphology of the particles, and chemical distribution. So I will follow with the boron doped hydroxyapatite at first, and if the time remains, I will mention about the silver. So for the boron doped hydroxyapatite powder, we focus on calcium deficiency and phosphate deficiency, as I mentioned, and as we expected, these two different deficiencies in the chemical solution cause different nucleation and different particle growth. As you can see on the TM image, the calcium deficiency causes rod-like particles. However, phosphate deficiency with the boron doping cause most like the needle-like powder morphology. And here the table shows the calcium phosphate ratio which fluctuates from the stoichiometric value. And also I also present here the content of atomic percentage of the boron content in the powder, which shows phosphate deficiency holds more boron into the structure. For the next, we characterize the phases of calcium deficient, which we call BC hydroxyapatite. With the increasing from left to the right, you can see we increase the boron content for the calcium deficient hydroxyapatite. And with increasing the boron content in the structure, we could see especially at the 1100 or 1300 sintering temperature, we could observe some decomposition of hydroxyapatite to alpha TCP or beta TCP. In generally, hydroxy was the main phase for all conditions. However, with the increase in boron addition and also with the increase in temperature, because of the increase in the crystallinity, we could observe the decomposition of hydroxyapatite to the calcium deficiency site. For the next slide, I show the phosphate deficiency hydroxyapatite with the boron incorporation. And in this case, same again from left to the right, you can see the increase in boron content and increase in temperature for each XRD figure. In this case, we only obtained single and stable hydroxyapatite phase, and there was no decomposition, which we concluded that this phosphate deficient hydroxyapatite resulted more stabilized hydroxyapatite structure with the special defect chemistry. And for further, we would like to investigate where the boron is and how the boron reacts in the hydroxyapatite structure. For this purpose, we took FDIR with the different boron content and with different deficiency sites, which BC is the calcium and BP is the phosphate deficiency site. As you can see from the FDIR result, we obtain the borate existence. We can detect the borate existence in the structure. And this borate existence, this borate positions gives us the idea of the borate actually incorporate into the structure or in the C-axis column. Depending on the position of borate peak, we can understand that. However, there is a difference between these two deficiency type, which one of the significant differences, the calcium deficiency show less decrease in the hydroxide density, and the phosphate deficiency, we couldn't observe such a behavior. And this shows the defect chemistry was different from the calcium deficient and the phosphate deficient synthesis process. And we would like to understand this deficiency. We would like to see the effect of this deficiency for the, of course, bioactivity tests. One other point to mention, one other important result to mention here is when we focus on the phosphate deficient hydroxyapatite, we could observe carbonated structure, which carbonated hydroxyapatite is well known with improved dissolution behavior, which dissolution behavior is also inductive quality of hydroxyapatite, which replace itself with the bond structure. So with these structural observation, we focus on how the osteoblast adhesion will result on these calcium deficient or phosphate deficient hydroxyapatite. And one of the observation we could mention is increasing the boron content on the structure. We decrease the grain size. We obtain decrease in the grain size, which decrease in grain size is also reported to improve the cell adhesion to the surface. However, if we can see the right side osteoblast adhesion figure 6, we can understand maybe the BC, which means calcium deficient tendency, increase with the increase in boron content. It might be the release of boron or the decrease in the grain size. However, the BP peaks has significant difference compared to pure hydroxyapatite, which shows the improvement, the enhancement of the osteoblast adhesion of the BP, which we discussed that it must be related to the carbonated hydroxyapatite formation, which improve the dissolution and, of course, release the boron, who show the pre-osteoblast activation, boron ions, and also hydroxyapatite dissolution as well increase the ionic concentration in the solution, which attracts the cells and improve their adhesion. In this boron concept, we concluded that phosphate deficient site incorporation improved osteoblast adhesion with the carbonated apatite formation. If I would like to continue with the If I can continue with the silver, which we focus on mostly antibacterial activity. However, before the antibacterial test, of course, we should understand the concept or how this silver doped hydroxyapatite form. After the synthesis, we obtain the nanoparticle attached with the spherical silver nanoparticle. We observe these spherical dark particle as a silver, metallic silver particle in the nano size, and they are attached to the hydroxyapatite particle. Of course, we expected that after sintering, the silver will incorporate it. Also, we had a report about this process as well. So with increasing the temperature, as you can see here, we didn't get silver, metallic silver peak, but instead, with increasing silver and temperature, we could observe decomposition of hydroxyapatite with the foreign ion incorporation. In this case, hydroxyapatite decomposed the beta TCP phase first, and then alpha TCP phase in the following heat treatment and increasing the silver concentration, which show similarity with pure hydroxyapatite from the XRD investigation. And this result is about the calcium deficiency side. So silver precursor replaced with the calcium precursor in the solution, in the precipitation method. In the next slide here, I would like to mention about the phosphate deficient, which silver exchanged with the phosphate side. And in this case, we similar to boron case, we could understand that phosphate side deficiency tries to stabilize the hydroxyapatite, but in this case, we still obtain a beta TCP phase decomposition. However, beta TCP phase decomposition still shows the enhancement of the stabilization of the structure, because in the high temperature of 1300 centigrade degree, beta TCP should transform to alpha TCP, like the calcium deficiency side. However, in this phosphate deficiency, it didn't decompose, and we could conclude this result as a stabilization of the structure in the case of silver to phosphate deficient side. When we follow the characterization of these sintered samples, and here is I show the sintered sample with the mapping with the characterization chemical composition. So we can see the particles, sintered particles still has calcium phosphate oxide hydroxyapatite structure, the elemental mapping, as well as the homogeneous distribution of the silver coming from this particle, which shows that it is not a cluster type, but it's also, we can say it's atomic incorporated to the structure, atomically incorporated. As well as the chemical distribution of the silver, we could also get the effect of silver to the structure when we exchange it with the phosphate, which phosphate deficiency P2AgHA from the FTI result, we can say that we have carbonated hydroxyapatite formation with the P side deficiency, which I also did mention about the boron side, boron case. The carbonated hydroxyapatite has improved dissolution quality of hydroxyapatite. And we also expect the result of carbonated apatite to improve the bacterial ratio. Also, the figure 12 shows this expectation comes true, which P2Ag is a carbonated hydroxyapatite compared to C2Ag, which they're all having, they both have similar amount of silver in the structure. However, the P2Ag shows significant difference to pure hydroxyapatite in the case of bacterial ratio, which we could conclude that the carbon hydroxyapatite formation improved antibacterial activity. In the other case, for osteoblast adhesion, for the silver case, our concern was the toxicity of silver, which can inhibit the cell adhesion and cell growth. But in this case, we didn't see any toxic effect from the calcium deficient and phosphate deficient sample, which we can conclude that there is no cytotoxic effect, and as well as improvement in antibacterial activity, propose this material as a candidate for bone graft or even coating material. So I would like to conclude my talk with the short and right to the point remark, which we fabricate the hydroxyapatite with boron and silver, with changing the deficiency site with calcium and phosphate, and the defect chemistry that we cause with the changing the with the precursor site in boron doping, enhanced osteoblast adhesion significantly. In the case of silver doping, we improved antibacterial activity without any toxic effect. Thank you for your attention. This is end of my talk. Thank you.